love breakfast. Professor Evil Scientist!
Crazy people, caramel luncheons and white milk.
Welcome to Aurora Public Radio's 6th entry in our ongoing series on biotechnology in honor of our ailing superhero, Biotechnology Man. Today's topic is animal biotechnology, a growing field in science concerning the manipulation of what are commonly referred to as genes is the very essence of biotechnology. There are many fields of biotechnology, but one of the premier fields is animal biotechnology. Many processes, from microinjection to embryonic stem cell gene transfer to retrovirus gene transfer, are methods of performing gene transfer in animals. Transgenic animals such as mice, sheep, pigs, goats, and even cows serve as great models for genetic manipulation and research. Research is being done on pigs for the potential successful xenotransplantations to prolong human life. In addition, the science of biotechnology is researching potential cures for debilitating animal diseases such as foot and mouth disease, mad cow disease, coccidiosis, trypanosomiasis, and others. And just when you think that that was all, animal biotechnology can also assist with animal propagation by methods such as artificial insemination and animal cloning. Check back tomorrow for our piece on marine biotechnology. Thanks for listening. This is Dan Moreland for Aurora Public Radio. Stay tuned for some more news about MRSA.
Good morning and welcome to Clone a Pet. How may I help you?
It's my mouse. My mouse died.
Oh my goodness.
Just made me I knew Trixie. She was my best friend.
Okay, yes, just have a seat. We can certainly do that. I'm just going to tell you a little more about cloning so you're more familiar. There are two different methods of cloning, embryo splitting and nuclear transfer. Embryo splitting is used to form clones. The eggs are fertilized in vitro, and when they're ready at a specific stage, they are separated from one another and transplanted into different mothers. This produces two superior animals as opposed to just one. One twin can be used for experimental, and the other one can be used as a control. It is also used for human infertility. However, you don't really need to know that because we're just worried about the mouse Trixie. Anyway, the second method is nuclear transfer. They increase the number of offspring to hundreds or even thousands of individuals. The first successful animal cloning by nuclear transfer was in 1986. This involved using sheep eggs. The sheep eggs were collected, the nuclei were removed, and each enucleated egg fused with a cell from a 1632 cell embryo. The fused cells were grown into embryos and transplanted into surrogate mothers. So a 32 cell embryo can give rise to a 32 animal clones. The first cloning experiments were done in 1950, and they were conducted by Briggs and King, and they were doing those studies to look at the differentiated cells of the leopard frog. And what they found is that as cells differentiated into older embryos, they were less likely to develop into frogs. Also in 1996, Scottish scientists Wilmot and Campbell at Roslin Institute produced a clone of an adult Dorset sheep using a nucleus from a differentiated cell, also known as a somatic cell, rather than from an embryonic cell. They named the clone Dolly. I'm sure you've heard of Dolly. It was very publicized. Dolly lived for 7 years, but post-mortem exams showed her chromosomes to be aging prematurely. She had arthritis, and she ended up dying of lung cancer, which was virally induced. There is a lot of controversy over Dolly. The controversy was over whether she was generated from a mammary cell or a stem cell. Also, in 1998, scientists cloned more than 24 mice using direct injection of nuclei into enucleated eggs. Non-human primate cloning is being worked on in other endangered animals also. So it should not be a problem to clone Trixie because there have been mice successfully cloned. And there's only one thing though, the cost is about $30,000. But if you're ready to take the plunge, you can sign here. And we will need a 50% down payment, and that'll be approximately $15,000.
Oh, anything from Trixie. Thank you so much.
You're welcome, and thank you for stopping by, Mr. Smith. And we will contact you when Trixie 2 is ready.
Thank you.
You're welcome.
Have a good day.
What is it? I'm very busy.
I'm sorry to interrupt you, Professor Evil Scientist, sir, but we finally have enough money for your evil plan.
Ah, yes, finally. With biotechnology man's heart's failing, thanks to my fracture wave, we can finally be able to get underway our plan.
So what do we do now, sir?
Transgenic cows are our plan. Transgenic cows is our plan.
Wow.
They've been experimenting in this field for about 20 years now, using gene for the past 20 years now, using gene knockout technology, which is replacing some genes, adding in others, and taking out others. We can replicate and process more desirable traits, such as an increase in production and make more faster, more powerful cows. For the past 2 months now, I've been collecting cow embryos. With these, they are fertilized in vitro on petri plates. I've been maturing them in petri dishes. With this, we can then fertilize them, and then we can separate the yolk from the pronuclei, with the pronuclei separated from the pronuclei. The DNA are then microinjected into embryos, and then the embryos are injected into surrogate females, which will produce cows at a much faster rate. And anyway, with all this, I have located genes in cows that code for intelligence, obedience, and kung fu. With those that know kung fu, and they know kung fu. With this, we'll be able to take over the White House and take over the world.
That's awesome. Very evil. Very evil.
Very evil. Very evil.